finish up chapter 12 on biotechnology. We're not really going to get into genomics. I did have a question on biology bites today about exam material. It is chapters 9, 10, and part of 12. Anything from lectures fair game, anything from the textbook is fair game. Again, most of the time, we focus on these learning objectives. So today only has 2 learning objectives, PCR and gel electrophoresis. Those are definite things to study for the exam, but that doesn't mean you should ignore everything else. Cole, the test opens Tuesday and due by Wednesday. So the test technically is on Wednesday, but I give you 24 hours. You're welcome. So again, be aware when you're studying about that. Homework 11 is on chapter 12 because we skipped chapter 11. So we didn't do chapter 11. There is no homework on chapter 11. So homework 11 is over chapter 12. And then we're going to go chapter 13 and then skip ahead a lot after chapter 13. I should stop, give time for any more questions. Good. Keep in mind, we only have about 3 weeks till Thanksgiving. And then 1 week after that, that's Dev Week. And then finals. Your final exam is not cumulative. It only covers 3 chapters. So your final exam only covers 3 chapters, just like every other exam in this class. Or almost every other exam. Last Wednesday, I covered gene editing and GMOs. Today we're going to get into DNA profiling. So polymerase chain reaction. So we talked about how cells make more DNA. Well, if I'm in a laboratory and I want to make more DNA, I use something called polymerase chain reaction. Polymerase chain reaction basically makes copies of one portion of the DNA, a small segment. It was developed in 1985 by Carrie Mullis and other individuals at the company where he was at. The technique has vastly improved in the last 35 years, but the basic principle remains the same. You're making copies, you're amplifying your DNA of interest. It's based off of DNA replication, a lot of the same concepts. So I do recommend looking at DNA replication and then comparing it to PCR. So what's different? Well, one of the things that's different is something called primers. During DNA replication, your cell makes primers. However, when you're doing PCR, you have to add primers. Primers are pieces of DNA that match whatever sequence you want to amplify. So if I want to make copies of the insulin gene, I would have primers that match or complement this end of the insulin gene, and I have a primer complement this end of the insulin gene. So I'm going to need 2 primers on either end of the gene I'm interested in copying. The primer at this end will complement the bottom strand. The primer on this end will complement the top strand. DNA is double-stranded, so you have a primer for each strand. Having a primer at the beginning and a primer at the end is called flanking. You're flanking your gene of interest by having a primer at this end and a primer at this end. Now the primers will cause DNA synthesis to go towards each other. I'll show it this in a video. Videos make this much more understandable. But literally for the primers, I say, oh, I'm interested in this gene. What's the sequence? Okay, type in the sequence into a company website, and 2 days later, I have primers in my mailbox. So you literally just buy the primers with whatever sequence you want. So there's 3 main steps to PCR. Denaturation. The DNA has to be separated, the 2 strands. Denaturation, in a cell, you use DNA helicase. For DNA replication, you have an enzyme that does this. Well, in PCR, denaturation, the separation, is done with heat. So step 1 is heating things up to denature, to separate your DNA. So step 1 is heating it up to denature your DNA so the strands are separate. Step 2, annealing. Annealing is when the primer binds to your DNA. So annealing is simply, you'll cool it down until your primer attaches to your DNA. Step 3 is actually making more DNA. DNA polymerase goes in and makes a copy of your DNA. This is the exact same DNA polymerase that's found in replication, but we typically use DNA polymerase taken from a special bacteria. The bacteria was originally found in Yellowstone National Park. Yay, national parks. And this polymerase is like super hardy. It can survive harsh conditions. And what makes polymerase special, or PCR special? You repeat these steps over and over and over again. So you do step 1, 2, 3, 1, 2, 3, 1, 2, 3. And you repeat it about 25 to 30 times. Now, fortunately, the scientist doesn't have to stand there and do 1, 2, 3, 1, 2, 3, 1, 2, 3. There's a machine called a thermocycler, or a PCR machine, that does it for you. So I literally mix everything together, stick in the machine, and press start. And walk away for 2 hours. That's it. I mentioned this DNA polymerase is special, and it's called TAC. TAC stands for Thermus aquaticus. That's the bacteria found in Yellowstone. And it can survive really high temperatures. So the DNA polymerase TAC can survive very, very high temperatures, and that's why it's important for this. Yes, sweet.
What are these copies of DNA used for?
It depends on what you're doing. So like, for instance, the recombinant insulin I talked about last time, I can make copies of that human insulin gene and stick the copy in E. coli, and now my E. coli can make insulin. Sometimes it's used in forensics. We are at the point, let's say Dr. Nesbit sneaks into Fort Knox, and I steal some gold, sneak out, and I leave 1 skin cell. They can use PCR on that skin cell, amplify my DNA, and find out who did it. Now, granted, for the average crime scene, they don't do that. But it's Fort Knox. We know they're going to do that. But yeah. So anything from forensics to recombinant DNA technologies to Dr. Nesbit's advanced molecular class does this to study bacteria. Here's the steps right now. So your sample DNA. This sample can be from anything that has DNA. You can take it from plants. You can have sample DNA from people. You can have sample DNA from bacteria, viruses. In fact, the COVID-19 test, where they swab your nose and it takes like 2 days to a week to get your results, uses PCR. And so for the COVID test, your sample DNA is virus. What viruses did they collect from your nose? you have a target sequence. So we know that we want to look at this one gene in the virus. Step one, separate the DNA. And this is using heat. Where's my annotator? So heat is what separates the DNA. Step two is annealing, where you have your primers, pieces of DNA. There's a primer on each strand. So the primers attach, they anneal. And then finally, your extension is when DNA polymerase actually makes the new DNA. So this process simply makes copies of DNA. And you might say, why do I want more copies of virus DNA? Well, we can't measure a few viruses like easily. But if you make copies of it, it becomes very obvious. So you can detect very, very small amounts of virus. Just like you could detect Dr. Nesbit's one skin cell in Fort Knox. All my students are going to be like looking out for me to break into Fort Knox now. It lasts like all of like just
uses repeated cycles of heating and cooling to make many copies of a specific region of DNA.
Okay, go back. Okay.
Chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA.
So they're heating it up.
First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the prime sequences and adds nucleotides to extend the second strand. This completes the first cycle.
Step one. Step two.
In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
So you can go from literally one single molecule of whatever you're interested in to 1 billion copies in about 2.5 hours. And all I had to do was mix it up, stick it in the machine, and press start. Now, you're like, oh, it only takes Dr. Nesbit 2.5, 3 hours. Why does my COVID test take 2 to 3 days? Because they're doing hundreds, if not thousands, of tests a day. And so they have to get to your sample. There's also a few extra steps in the COVID test because COVID is caused by an RNA virus. And you have to switch the RNA to DNA first. Then you do PCR, and then it checks for the, do you have it? So it's a few more steps. Advantages. You can take a tiny, tiny sample, literally a single cell or a single virus, and make lots of copies. It's fairly rapid. I mean, yeah, this is why, you know, for the people who are high risk or something, like President Trump, when he got sick, I'm pretty sure it only took him 2 hours to check him. He probably didn't have to wait for every other person to get tested first. It's also very specific. If Dr. Nesbit is sick with rhinovirus, it doesn't show up on a COVID test. because COVID is caused by a different virus, that like a coronavirus, and Dr. Nesbit has a rhinovirus. So you literally can tell the difference between this virus and this virus, or even you can tell the difference between Dr. Nesbit and my big brother. My older brother would show up different than I would if you have the right parameters. So it's very specific, pretty darn fast, and you can use a very small sample to get lots of copies. We have actually sequenced DNA from mummies because you need a single cell. You can get it from woolly mammoths that are 40,000 years old, plants that are 30 million years old that were fossilized. And I mentioned crime scenes. Blood has DNA in it. Skin cells have DNA in it. Semen has DNA in it. Typically for the rape kits, they're looking for semen, and then they can isolate out the DNA from the semen and find the person who committed the crime. Next fall, I'll probably be teaching forensic biology again and go into more detail about how those tests work. So Trent's asking whether DNA polymerase is like a zipper. DNA polymerase is more like the seamstress. So DNA helicase or heat acts as a zipper. So they separate the strands. DNA polymerase would be your seamstress who makes DNA. So the DNA polymerase literally makes more DNA. So PCR in 35 years has literally revolutionized our lives. Everything from like the Golden State Killer, it was someone who terrorized people all along California coast. And he got away with it for a long time. And he was later found through PCR and DNA sequences. And that was only a couple of years ago. So it's played major roles throughout modern times. Okay, I now have 20 gajillion copies, a billion copies, whatever, of my DNA. That's nice. Yay, I have lots of copies. Now I need to separate it out. Gel electrophoresis, you're welcome, Trent. Gel electrophoresis really is, how am I going to separate DNA from other DNA or proteins from other proteins? So when I say macromolecules, I'm usually talking about DNA, RNA, or protein. So there's many different versions of gel electrophoresis. Some do size, some do charge, like positive, negative, some do shape. For this class, oh, come on. Why does it not like me? We are only going to talk about size. If they're asking what separates, or it's based on size, and then we'll talk a little bit more about electrical charge. On the next slide, I'll talk some more about electrical charge. For this class, we're not going to get into separating based on shape. That's something we cover in upper level classes. So, separating them based on size, you basically make what's called a gel. And it feels kind of like gelatin if you actually touch it. And it's pretty solid gelatin, like you have to try to kind of break it. And there's little holes along the top called wells. And you stick your DNA in the little hole, and then you add electricity. Whenever step 2 says current is applied, it's literally adding electricity. Well, if you've ever looked at a battery, which I'm pretty sure everyone here has, you have a plus and then a blank. So here we have a positive end and a negative end on these gels. Shorter pieces will move faster. Shorter pieces move faster because the gel acts like an obstacle course. If I take the lecture hall and I put strings back and forth all over the lecture hall until it's a maze of strings, we get my 8-year-old daughter, who's not even 4, just barely 4 foot. We get my 8-year-old daughter in here. And then we get my dad, who's 6'10". And we say, get through this maze. Who's gonna get through faster? Who's gonna be able to go through those little ropes faster? The 4-foot person or the 6'10" person? Give you a hint, my dad's not gonna be able to get through that maze. So the smaller fragments, the smaller pieces of DNA go faster. And then you use fluorescence or radiation to actually see the DNA. And you'll see what's called bands. And every band is a certain size. I'll show this on the next slide, but every band, the one that says band, is a certain size. So if you get my daughter's entire 3rd grade class in here, they're all about the same size. So here's a picture of a gel in gray. The DNA is placed in little wells. And this DNA is a mix of sizes. And you have your electricity with a negative and positive end. So this would be the positive end of your battery, and the negative end. And you run electricity through your gel. The DNA will move from negative to positive. This goes back to chapter 3. I know y'all love hearing this. The DNA moves negative to positive because DNA has negatively charged phosphate backbone. In other words, opposites attract. So negative DNA goes to positive electron. So the reason the DNA moves, is that the DNA has a negative charge. After about 40 minutes to an hour, sometimes you can get away with half an hour, your gel is done. Each one of these lines, each one of the blue lines is called a band. So whatever band is at the bottom, that's going to be the shortest piece of DNA. And then we'll have the next tallest piece of DNA, and the next biggest piece of DNA, until you get to really large pieces of DNA that can barely move at all at the top. So whenever you're looking at a gel, the ones closest to the start, closest to the wells, are going to be big, and the ones at the bottom are going to be small. And here they're showing an actual gel that's fluorescing under light. And you can see some bands are faint, some are brighter, and you have different sizes. And this is one sample. This might be, say, the criminal. This might be from the victim. This might be suspect 1, suspect 2, on down the list. This can also be used if I want to isolate out a certain DNA fragment or something. I'm gonna skip over that. Okay, so a zoom in, I mentioned the band, so each one of these lines is a band. Short moves fast, long moves slow. Opposites attract, so negative DNA is moving to the positive electrode on the gel. Covering forensics, so crime scene. This is the DNA you got from the crime scene. You typically will do a PCR to amplify it. So this step 2, this is your PCR to copy DNA. So you get your DNA from the crime scene, you copy it, and then you do your gel electrophoresis. So suspect 1, you get their DNA, you copy it, do PCR, and then you compare this PCR to, or I should say on a gel, you run this PCR on a gel and you run this PCR on a gel. And you're comparing these 2 lanes. And when you're looking at it, I'll go with it. Like this band over here doesn't match anything in lane 2, but it does match something in lane 3. This one is found in lane 2 and in lane 3. This one in the middle doesn't match either end. This one matches here. And basically, whenever they're doing this for criminal cases, you have to get a certain number of matches for it to be considered likely to be the criminal. There's a drawback. Identical twins typically have the same pattern. You can distinguish identical twins at the genetic level if you get really specific, but you have to do extra work. The standard process would not distinguish identical twins at once. But you can tell, like, these might be brothers, and you could tell brother A from brother B based on this method. So now we've caught the suspect, suspect 2. They're going up, to court, and the jury gets to decide if there's enough evidence. So here's things where when you actually look at this, like when the Bureau of Investigations does this, they have multiple markers. And basically they look at like the crime scene, what markers you have, versus this person was put in jail, Earl Washington, because he was at the scene of the crime, there was a connection. Once they did DNA analysis with the PCR and the gel electrophoresis, 16 and 17 don't match. 19 and 18 don't match. And you keep looking, that doesn't match. Well, there's one match. That doesn't match. So he has 2 out of 6 matches. It wasn't Earl. So Earl got released from prison. And then later they found that Kenneth Tinsley was the criminal, and they did DNA analysis. You see that all 6 match. With current FBI, they typically want, I believe, 21 matches to be considered likely to have done it. So the standard has changed over the years as we get more information. But this is only showing 6 matches, just to simplify it. Little bit quick on genomics. This is, if you look at an organism and you look at every single gene in the organism, And you look at how those genes interact. Initially, everything was done on prokaryotes because prokaryotes are simple. Prokaryotes are straightforward, they're small, they're easy. It was almost 10 years after E. coli was sequenced before we got a human genome. And the human genome was a rough draft, that first release, like a very rough draft. It's kind of like the draft you don't even want to show your friend because you're like, that's pretty bad. But they wanted to have something. In 2013, when these slides were first put out, there was 7,000 species. Yeah, there's a lot more than 7,000 now. When scientists sequence genomes, oops, there we go. If you have any money whatsoever from the US government, you must publish your genomes in a database that is freely available to everyone. So, stupid. Come on. I'm making noises at the computer. So, this is the United States National Library of Medicine, which is under the National Institutes of Health. And it's just showing genomes, genome information by organism. You can look at, so scientific names, it has the size of the genome, is it a chromosome or a plasmid? Does it have chloroplast or mitochondria sequenced? And there's 14,000 eukaryotes that have been sequenced. Some of these are similar to each other, like you might have, say, corn, sweet corn with feed corn, like both are sequenced and those would count as two separate things. Some of these are completely different species, like soybean. Plants are really, really hard to sequence. Really, really hard. It's much easier to do animals. So we actually have a lot more animal genomes than we have plant genomes. You'll also see bacteria in here, some viruses, and there's just a whole lot of stuff. Like the first thing they did after COVID-19 was identified was they figured out which virus caused it, and they published the sequence of the virus. We know the sequence of the virus, and it's published in here. And people have verified it. Severe acute respiratory syndrome, coronavirus 2, there's the genome. The size of the genome, it has one chromosome. And you can actually, is it this one, I believe? It's going to give me, so this right here is showing me the protein sequence, not the DNA, not the RNA sequence. But I can look at proteins in the genome. Here's another protein in the genome. And these proteins are the proteins that attach to your cell or help the virus replicate inside your body. And so then it can be used for drug studies. So all of this information is available. And this is the original isolate from Wuhan that is completely sequenced. This virus was first published in February of this year. So we can sequence viruses very quickly now. And so you can go to this website whenever you want and look up your favorite organism and learn more about it. Year completed, the first bacteria was in 1995. I said the rough draft was in 2000, the actual completion was in 2003, the actual good one. And notice there's, there's one plant done early. Haploid genome. So humans actually have 6 billion bases. So like Dr. Nesbit, one cell would have 6 billion bases. But for simplicity, because some things are haploid, some are diploid, some are tetraploid, they just said, okay, we'll say if you only have one set of genes, how many would you have? And so this number, 21,000, actually changes. Don't try and memorize this because people are still arguing over how many genes people have. But the things you can tell from this, more complicated organisms typically have more DNA and more genes. So if you compare us to yeast or E. coli, we have a lot more genes than either yeast or E. coli. Go figure, we're more complicated. Makes sense. Now, what they're not showing, there's actually plants that have way more genes than humans. And so that's something else. This also helps us, chapter 13 will be on evolution. So a segue into evolution, you can look at how things are related in the evolutionary tree through genomic studies. You may have heard the thing that says, oh, we share 96% similarity with chimpanzees, humans too. And the reason why this is of interest isn't to debate human chimpanzee relationships. It's also that, hey, that yeast over there has the same genes we do. Some of them, obviously not all of them. I'm not a yeast. Well, if that gene malfunctions in the yeast, can that tell me how to help treat the human? And so a lot of medical studies, you're not going to go start fiddling with human DNA. You're going to go study it in the yeast. You're going to go study it in the lab rat and that sort of thing. So that way you can learn how things work in lower organisms like yeast. And then once you know what's happening, say, okay, is that the same that's happening in humans? How can we make people better as far as medical treatments for immune disease or other factors? One of the cool things is I have someone I know has genetic disease in their mitochondria, and they know how to treat it because yeast have mitochondria. And they're like, oh, yeast who have this defect, you give them extra folic acid, they're healthier. So you, let's give you extra folic acid, it'll make you healthier. Evolutionary relationships. So these are DNA sequences from different sea slugs. So each one of these, this is a genus, and then on the right is a species. So you have 5 different species of sea slug, and each species has a different DNA sequence. So the Leonia would be this first sequence, the Araneae would be the second sequence, Digitata would be the third sequence. And you can see these sequences at the beginning, that's identical, but at the third position, there's some differences. As you work your way down, you count how many things are the same and you count how many things are different. The closer they are, the more they're the same, the closer relation they are. So like, they could be siblings, cousins, second cousins, whatever, fourth cousins, or completely different. You can figure out how far apart are they in the family tree. Human Genome Project. This is actually ongoing because while we have the sequence for humans, that's a composite. It doesn't mean that we have the sequence for every human. And so while we know that people have, say, red-green site the gene to be able to distinguish red from green on the X chromosome. There's other genes we don't know where they're at. So, for instance, try to think of, there's like a good one would be ADHD. We know that ADHD has some sort of genetic component, at least some forms of ADHD. And we know this because if the mom has it, there's a certain likelihood that the kid will have it. It tells us it's genetic. But we don't know what gene it is. Or it could be polygenetic. It could be 5 different genes. So you have everything from mild ADHD to severe ADHD. Severe might have 4 of the 5 alleles that cause ADHD, and mild might just have 2 of the 5. And so part of the Human Genome Project is figuring out, hey, where are these genes at? So while we have the sequence, we now need to figure out which genes go where. What do they do? Okay, I'm gonna go ahead. That's my last slide. And again, your exam is on Wednesday. This is exam 4 on chapters 9, 10, and 12. So I hope everyone has a good day, and I will see y'all on Friday.